Hey everyone. Today in Lesson 13, we're going to be discussing infectious diseases and epidemiology. So, in my background, I've worked tangentially on a lot of different infectious diseases because I worked as a vaccinologist for a time. So, I know a lot about various different diseases, their pathology, what we can do to stop them, what we can do to treat them, and we're going to dive into that pretty deeply in this lesson. So, there's several different types of infectious diseases that we can deal with. There is communicable diseases. Communicable means it can be spread person to person, and this is the majority of the diseases we think about. So stuff like the flu, whooping cough, even STDs, for instance. Then there's non-communicable. This is something that's acquired from the environment. Now, it's not to say that some non-communicable diseases might not pass if you share like a blood donation or something like that, because if you get infected with something, it's in your blood. You could give it to someone else and it could infect them as well. It's just saying that these aren't typically going to be spread person to person. So person to person contact is communicable disease, and then environment to person contact is going to be non-communicable disease. These are things like tetanus. So if you get cut on a really rusty piece of metal, you need to go get a tetanus shot, a tetanus booster, because there is a bacteria tetanus that lives on rust, essentially, and that, when it infects you, can have some horrible, horrible symptoms and lead to death. Similarly, valley fever is something that's just in the soil that we can end up breathing in and infecting our lungs. So, those are a couple examples of non-communicable diseases. Now, there's also two different kinds of pathogens that fall under these infectious disease umbrellas. There's primary pathogens, which cause disease in a healthy host. This means that they infect you and they replicate, and you are the normal host that they are trying to infect. Though that's the normal area. So the lungs, in the case of something like tuberculosis, that's what it's trying to infect, those exact cells in your lungs. And this is what is going to typically fall under disease. But then there's also opportunistic pathogens. Opportunistic pathogens are normally harmless, but in different scenarios, they can cause disease. Usually, this is someone who is immunocompromised, or you get an exposure from something that typically your body would fight off, but maybe you just had the flu, maybe you're low on certain vitamins, and for whatever reason, it's able to actually get in and infect you. An example of this is a lot of skin infections, like outer ear infections or just normal on your skin infections, come from bacteria that are typically just living on your skin in a natural state. But when a wound is opened, it can enter in and it can start to fester and replicate and actually become this opportunistic pathogen. Now, we've talked previously about the different periods of a disease. So, the first period is going to start with the incubation time. This is when the pathogen is present, but you don't have symptoms yet. So, someone, sneezes on you, right as you're walking through the airport, you get on the airplane, you fly, you're 2 days into your vacation, then you start having symptoms. Well, that first period is an incubation period. Going off this example, then you start having some symptoms. You're not really sure what they are. You have a little bit of a headache, you're feeling a little tired. This is the prodromal phase. This is early non-specific symptoms. Typically, it's going to include fatigue or malaise, is another term that just means you're feeling generally not well. After that, you might start having a really runny nose, a fever, a sore throat, a cough. And that is what we call the actual period of illness. That is the period you're in where you're exhibiting the illness that would be classified under a specific infectious pathogen. Then after that, you start feeling better, but you're not fully better. You are in this decline phase where you are not declining, you're actually feeling better, but the pathogen number of pathogen replicates inside you is declining. And this decline in the pathogens leads to you feeling better, the fever breaks, your symptoms subside, and the pathogen is going down, down, down. Then there's convalescence. This means that you, as the host, are returning to your baseline health. So you are entering this period after being sick where you wake up one morning and you say, I feel totally normal. I slept a full 8 hours. I'm feeling great. And you resume your daily life. That's not to say that there's no pathogen left. It's just it's such a little amount and your body's going to continue fighting it down to nothing over time. And you feel as a whole human totally better. And this kind of follows an acute disease trend where you have pretty quick incubation. You start feeling bad in that prodromal phase, then the illness happens, and then the decline where you feel better, but the actual virus is getting cleared happens, and then you go to convalescence where you return to your baseline. But it can also happen in a similar way, but in chronic disease. So you have longer incubation periods, sometimes years. Then you have a long prodromal phase where you just feel terrible for sometimes again years, and then you enter an illness phase where you're having more acute, more specific symptoms. And an example of this is hepatitis. So hepatitis is a disease that when you get it, it causes cirrhosis of your liver, so it's very, very bad, but you don't catch it for a very long time. And tuberculosis is similar for something that infects your lungs. You might have this long period where you're coughing, you're wheezing, you don't feel good. If you go on a run, you get tired more easily, and that might track for months or years before you actually start having the symptoms we associate with tuberculosis. And then latent disease is something that you enter that convalescent stage, you feel better, but then it can reoccur after a period of time. This is herpes or shingles, so something that can lay dormant and then pop up and have this cycle of disease once more. Now, when we think about disease, there's a lot of important factors that go into how bad the disease is for the person we're looking at. There's something called pathogenicity. This is a microbe's ability to cause disease. Realistically, we have E. coli living in our gut at all time. But if you were to take a, gallon of E. coli and drink it, you're still going to get incredibly, incredibly sick and probably, honestly, overload your body and die because of the amount of pathogen in your body, even though it might have a low pathogenicity. So the ability to cause disease is not the ability to make you sick. It's the ability of a increasingly small number of pathogens to make you increasingly sick. This is why things like anthrax and tularemia are very dangerous diseases, and you'll hear about them in, discussions about biosafety, bioterrorism, because a very small number of those pathogens can cause many people to have disease because it has a very high pathogenicity. Then when we talk about those opportunistic pathogens, stuff that's living in the soil, you can inhale that all the time, but you need enough to actually cause disease. So those have a very low pathogenicity. Then there is virulence. This is a degree of pathogenicity. So when we're talking about pathogenicity, we don't normally refer to it as that. We refer to it as virulence. And this is just the amount of people that can be infected by a certain amount of that bacteria or virus, whatever the pathogen is. And this gives us two interesting things that we typically look at in a lab. One is ID 50 or infectious dose 50. So that's where the ID comes from, is the infectious dose. This is the number of microbes needed to cause infection in 50% of the hosts tested. So if you have 10 mice and you inject them with, I don't know, 2 units, let's just call it, of anthrax, it is the amount required to infect 50% of that group. Then there is something called LD 50 or lethal dose 50, and this is the amount of that pathogen that's required to kill 50% of the host. So ID 50 is always going to be lower than LD 50, but sometimes these are very, very close in very dangerous pathogens. So botulism toxin is very, very infectious. So it has a low ID 50, and then it also is extremely deadly. So it has a low LD 50. And those two things together make it highly virulent, which means it has a high pathogenicity. I know that's a lot of terms to take in, but it's just getting down to there's these two tests that we do that tells you how dangerous that specific pathogen is. Now, when we talk about epidemiology or the tracking of disease, there are specific steps that occur when doing an epidemiological tracking of disease versus a tracking in a specific person. So when we're looking at pathogenesis, so how did this person get infected? We are going to look at their exposure. Where did they contact and encounter the microbe that infected them? The adhesion is also an important factor. So that's how easily the microbe can infect a person. Usually, this is done by testing tissue samples. So, how easily can the microbe infect that tissue? How much of that microbe is required to infect the tissue? How easily it is able to evade our defenses and spread. So, this is an invasion, is what we call it, the microbe's ability to invade and get through our defenses, infection, actual colonization and damage occurring, and then transmission. How does that pathogen leave the host to affect other hosts? And this is kind of the crossover between lab study and going into that epidemiological study. You need to understand all these things. Mostly on the research side, we're worried about adhesion, invasion, infection. So, how does this microbe actually function? Whereas on the epidemiology side, we're worried about exposure. How do we get exposed? With COVID-19, we know it's through someone coughs and it releases micro droplets, and the COVID is in that. And in that breath, in those micro droplets, we can get exposed because it's what we call aerosolized. It's in the air for that moment. But there's other things like HIV that's very, very difficult to contract from someone. Usually it takes sharing of blood or sexual intercourse to contract HIV. So that's what we're worried on the epidemiology side is that exposure slash transmission when we're looking at these things. Now, going fully into epidemiology, it's really important that we track diseases. It's important for deadly outbreaks, something like Ebola or Marburg virus, which kills the majority of people that are infected, 50 to 80 and sometimes even 90% of people who are infected, that we can look at these. We know the transmission. We know what the different contact events are. We know the incidence. So the incidence is the number of new cases in a time period because these diseases exist in the world. And if one person contracts Ebola and dies, that's worrying, but it is a low incidence rate. And that's probably the only person that's going to die if no new cases pop up in a short period of time. But if all of a sudden 50 people are going to hospitals with Ebola, we know that we're going to have a huge problem because of that incidence rate, the number of new cases in a time period. Then there's also prevalence, which is the total number of cases. So this is not new cases, this is the total number, the new plus existing. So we'll talk about things in epidemiology like the incidence is 500,000 people and the prevalence is 1 million people. So that means when we're looking at incidence rate, say, of 3 months, we've doubled the number of people infected in 3 months, and we're looking at a time course of, you know, 9 months. So in that first 6 months, we got 500,000 people. In this next 3 months, we have another 500,000. So this is ramping up. This is going to be a problem versus if the prevalence is, 10 million, and in the last 6 months, we've had those 10 million. And now in the last 3 months of that prevalence, it's only 2 million. So 8 million happened in the 6 months prior, 2 million happened in the next 3 months. That means we're doing a good job of controlling this infection as it attempts to spread. Now, this leaves us to some patterns of occurrence that are the typical way that you will find clusters of disease in a real world scenario. There's sporadic, this is occasional, kind of random, and this would be something like tetanus. A lot of people are vaccinated. Sometimes someone will not be vaccinated, they'll get cut, they'll come in with the disease. Maybe they're immunocompromised, maybe they're on chemo, maybe their, immune system just has a faulty gene in it. Whatever's happening, they come in, but that's sporadic. We know this is a fairly well-controlled issue. Then there's endemic. This means it's constantly present. So valley fever is endemic in the Southwest. you might contract it here, you might get sick with it here, but it's not spreading outside of the Southwest because you have to be exposed to it in the soil, essentially. So it's endemic. Then there is an epidemic, which is a sudden increase in cases that is within a singular region. So usually this is going to be something like continental or even country based. So you might have an epidemic in uh, the United States, you might have an epidemic in Canada, you might have an epidemic in North America, for example. And this is something that is, an E. coli outbreak. There's some food that is incredibly infected with E. coli. We're having an outbreak right now. It's so bad, we call it an epidemic. And there are very specific reasons and checklists for calling something an epidemic, and the same with a pandemic, so a global spread of a disease. And a historical example of this is the plague. A modern example, of course, is COVID-19, so something that's spreading globally. Now, there's a lot of characters in epidemiology. Uh, I don't know why I say characters, a lot of historical persons in epidemiology that have given a lot to that study. One of them is Robert Koch, who you've heard about in previous lectures and lessons, and that is because he found that pathogens are involved in all disease, but they are absent in healthy people. He isolated pathogens and grew them in pure culture, meaning he was able to actually take someone's sample who was sick and isolate the pathogen causing the disease. He then took healthy subjects. Of course, these were not typically people, and then he would expose them to the culture. This would be like mice and guinea pigs, and then those animals would get the disease, and then he was able to re-isolate that same pathogen from the new host. So he's someone that traced this. Okay, we have something infecting someone. I can isolate that. I can re-expose someone to that, and then they will get that disease. So there's one thing causing one disease. Then there is John Snow, and he tracked cholera outbreaks in London, and he is attributed as the founder of epidemiology because he looked at a whole system, a whole area of London, and said, why are these people getting sick? And tracked it to cholera in Wales. Now, there's a lot of other people, obviously, who are involved in these things. Over time, it's a constantly updated field. We learn more and more about diseases and how they spread and what's involved in that. There's people who study what's called One Health, so that is people, animals, and the world being usually agriculture, but the world as in, okay, if we're shipping, beef product from America across the ocean and we don't have really good checklists and refrigeration records, and then we feed it to people and they get sick, that cause is probably the refrigeration and the transport, not necessarily the meat itself, not the packing of the meat, it's the shipping, something along that line. And that's where this One Health comes in, that if we're all healthy, if we have healthy systems, that people will remain healthy and we can have a healthy overall world together. Now, there's a little bit more about disease I want to discuss, and this is how that transmission actually occurs in a lot of cases. So, if you're thinking about how things transmit, it's usually about going to be direct contact. So, person to person. This is something like sexual contact or those droplets that spread things like flu and COVID. But there's also indirect contact. A lot of bacteria will live on things like doorknobs or washcloths, and that's why we use paper towels and air dryers nowadays because we learned that having people use the same towel over and over again in the bathroom when they maybe didn't wash their hands very well could actually spread disease versus reduce disease. There's what we call vehicle transmission. This just means that it's being transmitted with something that we are taking in, whether it's food, water, or air. And then the last one is vector-based transmission. This is usually going to be biological, so something like a mosquito that infects you with its bite, giving you malaria, for example, or mechanical. And mechanical means that it is infecting you usually through a insect or worm or something like that, but it's not the direct contact with the biological unit there. It's not the contact with the fly or the worm. It's something else that it's carrying on it. The same way that bees carry pollen, a lot of other things carry pathogens with them. So flies can carry different pathogens just on their feet. They're going on all sorts of surfaces, and that can transfer if it lands on an open wound, for instance. Now, when we talk about disease, we also need to talk about one more thing, which is emerging and re-emerging diseases. There are many, many diseases that have been around with us basically forever when we try and track it in the genetic history of humanity. One is Epstein-Barr virus. Epstein-Barr virus causes infectious mononucleosis. It can cause different kinds of cancers. It is highly linked to causing MS and some other diseases, but we evolved with it, similar to a lot of the different herpes that we have. So there's herpes that affects basically every single living organism. There's herpes in seals. That's actually a big problem because they can die from it. But we've co-evolved with these, and these are what we would call re-emerging diseases if they were previously controlled and are resurging. So they are diseases which have been around for a long time, but something like a mutation or something else is causing them to re-emerge above the typical baseline in the population. An example of this is tuberculosis, just because it is becoming more and more resistant to our treatments. So it is continuously becoming more and more of an issue. And then also measles, because people aren't getting their measles vaccinations. That's trending downwards, and the measles vaccine is not a lifelong one necessarily. So there's some people that are even vaccinated, but it has waned over time. So they come into contact and they get measles. And this is a huge issue because it is a pretty deadly disease for elderly people and then also, of course, young children. And we had it under control, but now it's coming back. And this is very different from emerging diseases. Emerging diseases are brand new. So, an example of this is Ebola. Ebola is not too old. It's estimated it's around 50,000 years old and came out of a part of Africa. I do forget which off the top of my head, but it evolved and emerged, probably came from bats or another zoonotic uh, carrier. It evolved there and then jumped to humans. And now it seems like it will one day be solved, but right now, if you get infected with it, it's a huge issue. There's not a lot of treatment options. The same thing with Marburg and also COVID-19. COVID-19 was this emerging disease that we had studied, we'll call cousins of COVID-19. So SARS and MERS, which were much more deadly actually, but can't spread well because of how deadly they are. And then COVID popped up and it's in that same family and it started infecting people and kind of running amok, as I'm sure you're all too aware. And these emerging diseases are very dangerous because we don't have a good repertoire of treatments and protocols in the clinical setting to deal with these. Now, I know that is a lot of information. I think it's really cool. The life cycles of these different diseases really end up playing a critical role in the epidemiology. And hopefully, you're starting to see how all this microbiology coalesces into informing us not just how to treat an individual, but also how to treat diseases as a public health issue and tackle them in very simplistic ways that have huge impacts. Vaccination, for instance, uh, identifying emerging diseases so that we can develop therapies to them, having just the treatment of water be standard. So everywhere in America, for the most part, Flint, Michigan exists, and those places where drinking the water is probably a bad idea, but for most of America, you can drink the water. In other countries, they have less robust treatment programs for water, so you can get sick if you drink the water there because you're not used to those pathogens that are there. You haven't been exposed. So when someone got sick as a kid who grew up there because those pathogens is now immune, you're not, you go there, you get traveler sickness. is what ends up happening. So it's a very interesting time we live in because we all have all these amazing new tools. We have genetic sequencing of viruses. We have great screening programs worldwide, but it's also a very scary time because we're learning more and more that there's more infectious diseases than we thought. There's random things that could become huge issues, such as COVID-19 coming out of something like SARS and MERS, which luckily wasn't that deadly by comparison. And then uh, looking at the reemergence of diseases that have been pretty much wiped out for a long period of time. So it's an interesting time. It's sometimes a scary time, but that is the world of infectious disease. Hopefully, it was at least interesting. As always, if you have any questions, please let me know, and I hope you have a great week.